Hi again. 5-3 for you guys. Microscopic anatomy of a skeletal muscle fiber. So we talked about how the muscle is made up of bundles which have cells inside. And so inside of the cells, the fibers, remember, cell equals fiber. In this particular case, we're going to take a look at some of the structures inside of the cell. Hey, what's up? Oh, okay. Thanks. We're going to take a look at the fiber itself inside and all the cool structures inside. Okay, so let's see. Each fiber cell, oh, now it's really small. I don't like that. That's too big. Come on now. I just wanted up at 4. There, 4. Yay, okay, sorry. So each cell is a long cylindrical cell. So like I said, it looks like a giant pencil, like this. And all along it, we see nuclei. Why is there many nuclei in one cell? Because like I said, they used to be lots of different cells, but then eventually they got fused together and just became one giant cell. Now, if we take a look at it on cross-section, so if I'm looking at this edge straight at you, what ends up happening is that there's so much junk inside of the cell that the poor little nucleus is just crammed along the side. So the nucleus you can always see popping kind of out of the cell membrane of the cell because there's no room for it. It's too big and fat. So you'll always see like little bumps sticking along it, and those are all the cell membranes. Okay, now I had mentioned that there's tons of words that they use in place of what we're used to. So here's a couple of words you need to know. Sarcolemma is the same as the cell membrane of a regular cell. So, we draw cells, nucleus, and then we go, oh, there's a cell membrane. Well, here's the cell, here's all the little weird fibers and stuff inside, and that's a sarcolemma. Why they can't call it the cell membrane, I have no idea. Okay, then inside of the cell, here's a nucleus. We usually say that all this fluid in here is cytoplasm. Well, they can't call it cytoplasm. They have to call it sarcoplasm. Notice that word sarco means muscle. So we got sarcoplasm, which is the cytoplasm of a muscle cell. Not a whole lot of it. Why? Because there's too much junk inside of our muscle cells. So there's not enough room for it. Now, there's a special word here called syncytium or syncytium, depending on if you do the TI as a TI or a sh. So one fiber, because it's made up of several cells mushed together, they call that cell a syncytium, which is just a whole bunch of cells mushed together. And then inside of one fiber, so here's a little cross section of it, we will find lots of smaller structures called myofibrils. Now today I used my pencil as an example. So let's see. If I look on the edge of a pencil, okay, there's the wood right there. And then outside of my pencil is the yellow paint around the outside. And then inside is the lead. Okay, so can you see what you smell what I'm cooking? So the lead is a myofibril. But in this particular case, there are lots of them. And so each cell has several leads, several myofibrils, like this. And they are all tucked underneath the cell membrane. Now the cell membrane, the sarcolemma, is wrapped up by what? What is that you say? Yes, it is wrapped around by endomysium. That's a Y. Mysium. So it goes, the cell, cytoplasm, all that, then it goes the cell membrane, sarcolemma, and then it goes endomysium. Okay? Because remember we said that was the yellow of the pencil, so that's the outside. Okay, now, the sarcoplasm, cytoplasm, contains many glycosomes. That means glucose bodies. Basically, they're just big old packets of sugar, like the little bags you get to put in your tea. So, little packets of sugar because your muscles need lots of sugar to break down and make ATP. Also, lots of myoglobin. Myoglobin is just myomuscle, globin like hemoglobin. It carries oxygen. Oh, hey, look, I wrote the words right there. So glycosomes, granules of stored glycogen, so like little packets of sugar, and myoglobin, which is like hemoglobin, but it's found only in your muscles, and it just holds on to oxygen a little extra longer. Now, inside of the cell, we said there's the pencil lead, which are called myomuscle Myofibrils, fiber. So this is the pencil lead inside. It's an organelle, just like you've got a mitochondria, you've got a nucleus, you've got endoplasmic reticulum. Those are organelles, right? So this is just a funky new organelle that's only found in muscle cells, and you can have hundreds to thousands of them in one muscle cell, one fiber. So if here's my one muscle cell on edge, all of these little polka dots inside, those are all myofibrils. These are what actually make the muscle cell contract. Okay? All right. So here's a picture. So here is this outer portion, this little triangle and this part, sarcolemma, which is what? The cell membrane. Okay? Here's this poor little nucleus that's been shoved off to the side because he doesn't fit. And then we've got the myofibril. Okay, so this is the pencil lead. So this whole thing is the pencil. This is the outside of the pencil. Surrounding it would be endomysium. That's kind of surrounding it around there. Now in here you notice that there's bands. There's two kinds of bands. There's dark bands and light bands. The dark bands they call A bands. The light bands they call I bands. And that has to do with these little stripies right here. So we'll get into that in a minute. Okay, so we're going to go into the structure of a myofibril because that's the actual part that contracts in the cell, causing the muscle to shrink. All right, so this is one of the fibers. So this is my pencil lead right there. That's the one pulled. So if I grab one and pull it out of a cell, this is just one. And when I look at it, I can see it's got stripes. Remember stria, stripes. So striations are the stripes on the muscles. And all they are alternating dark and light A bands and I bands. So you can see right here, we have the A band, which is from here to here. So in your muscle, this appears dark. So that's the A band. Whereas the I band is light, and that's in between the two. So this line appears light, so that's the I band. Now within the bands, we have different zones. So in the dark band, the A band, we have this little stripe right here. Makes it a little bit darker. That is called the H zone. Well, it looks darker here, but it shows up lighter in the thing. So we have the H zone, which is made of special little filaments we'll talk about in a little bit. So it's a lighter section in the A band. Then we have what's called the M line. Okay, and the M line is, let's see, where is it? Right here. That's the actual little black lines. Okay, so you're thinking, what? What are you talking about? Okay, look at the blue. See these little blue lines? If I connect the dots of the blue lines like that, there, do you now see the H zone a little bit better? It's a little bit lighter, and in the H zone are these black things, which is the M line. I know this is stupid. Okay, it's confusing. It'll take a little while. Okay, and then we've got in the middle, in the I band, that's lighter, there's a little zigzag, zip, 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 all the way down, called the Z disc. Okay, that would make sense. Z, it looks like a Z. Makes sense. Okay, these are all the different bits and pieces of a myofibril. Remember, you got thousands of these per one cell. Now, let me go back for a second. From Z disc to Z disc, that is the functional unit of this guy called a sarcomere. So I'm going to take this chunk and make it a little bit bigger. Okay, here's a sarcomere. So those are the blue lines right here, and we can see they're much more complex. They're two fibers that kind of twist together. They're made out of actin. Actin is a really thin filament. So let me go back and show you. That's these little thin blue lines right there. That's actin. Okay. Then we have thick ones called myosin, and the thick ones are these big red ones right here. So let me go back. It's the red ones right there. Okay. And let's see. And then here's the Z disc. Bloop, 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 bloop. So we go back. Z disc. Oh, let me get rid of that. There. Bloop, 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 bloop. Z disc. Okay. So we've got, and there's our M line. Do you see the little dashes going down? So go back here. There's our little dashes. M line going down right there. Okay, so now you kind of see I'm trying to show you that they're the same. What we're not seeing are these little squishy curly things right here. They're lighter in color, and those we'll get to in a second is called titin. There it is. Titin. Not titin. Titin. Okay, so myofilaments are these three things. So 1, 2, 3. These are myofilaments. They make up the myofibril. Lot of words. Okay, so myofilaments make up the myofibrils. Myofibrils make up the fiber. Alright, so thick filaments, myosin, they extend the entire length of the A-band. So here they are. They're green now. We have the thin filaments, the actin, which go all the way across the I-band and somewhat into the A-band. And then we have these cute little things called titin, and these guys anchor the thick filaments together. They're not on this picture, so let me go back to the other picture. Hey, look, there they are. It's these little guys that go wiggy, 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 and kind of hold on to things. Wiggy, 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 right there. They hold on to stuff. They're springs. That's basically what they are, little springs. So titin are little springs, so we can't see them, but they're right in there. Okay, don't worry about this relaxation contraction thing. We'll get to that a little bit later. But I think that's enough to confuse you for now. Okay, so we'll talk about this tomorrow in class and go over it a bit. All right, see you guys later. Bye.